Everything you think you know about how prehistoric mothers fed their newborns rests on an assumption so quietly embedded in the history of science that almost no one has ever challenged it directly. The assumption is this: that breastfeeding in the ancient world was simply what happened. A baby was born, a mother nursed it, and that was the complete story. Simple, natural, inevitable. What 50,000 years of accumulated evidence actually shows, drawn from ice cores and bone chemistry and ethnographic fieldwork, and the lipid residues inside 3,000-year-old clay vessels, is something far more layered, far more collective, and far more dependent on the specific intelligence of women than any mainstream history of human survival has yet been willing to say plainly. Here is a number: 500 calories. That is the additional daily energy requirement of a lactating woman above and beyond her baseline metabolic needs, not 400, not somewhere between 3 and 6, depending on the study. 500 calories every day for as long as she nurses, and in the prehistoric world, that could mean 3 to 5 years per child. Now consider that the average daily caloric intake available to a woman in a mobile hunter-gatherer group in a non-tropical environment varied significantly with the seasons, potentially dropping to bare adequacy during winter months. The arithmetic of those two numbers, 500 calories required, uncertain calories available, is the arithmetic of infant survival in the ancient world. When the math failed, babies died, and the math failed with a regularity that every community in human history had to develop a systematic response to. Your own body carries evidence of that response, not metaphorically, structurally, the particular architecture of your immune system, the specific bacterial populations that colonized your gut in the first weeks of your life, the neural wiring that generates your involuntary response to a crying infant. Regardless of whether you are the child's mother, these are biological signatures of a survival system so ancient and so consequential that it shaped the female body at the level of evolutionary selection. You are not merely descended from women who survived these conditions. You are, in a measurable biological sense, a product of the specific solutions they developed. 3 to 5 days. That is how long a new mother must wait for mature breast milk to arrive after childbirth. In the meantime, the breast produces colostrum, a thick, dense, antibody-saturated substance that modern medicine considers nutritionally irreplaceable for a newborn's immune development. But across ancient Egypt, across medieval Europe, across dozens of documented traditional cultures stretching into the present century, colostrum was not trusted. It was believed to be harmful, possibly toxic, certainly inadequate. Mothers in these traditions were advised to withhold it, to give their newborns something else in those critical first days: honey, sweetened water, animal milk, herbal preparations, wine diluted with sugar. Every single one of those substitutes was demonstrably inferior to what the mother's body was already producing, and yet the practice persisted across continents and millennia with a consistency that tells us it was filling a real psychological need, even as it addressed an imagined physiological problem. Someone was always reaching for something to fill a newborn's mouth in those first hungry hours. That impulse ancient, cross-cultural, powerful, is the entry point for the entire story we are going to tell tonight. Because the real question is not what prehistoric mothers gave newborns in the first 3 days. The real question is what they did when the milk never came, or came and then stopped, or was never sufficient to keep a child alive through a hard winter. What did a mother do when she died in childbirth and her 3-day-old infant was left in a group of 40 people in the middle of a glacial plain 25,000 years ago, with no formula and no clean water and no medical guidance? Only whatever the older women in that group had learned and carried and passed forward. What knowledge system had those women built over the preceding generations, and how had they maintained it, and how well did it work? The answer, when you follow the research from its edges to its center, is one of the most sophisticated and consistently overlooked stories in the entire history of human survival. It is a story about collective intelligence held entirely outside institutions, outside writing, outside any of the formal structures through which knowledge is typically preserved and transmitted. It is a story about what women knew, what they invented, what they borrowed from each other across communities and generations, and what they paid the cost of when they got it wrong. It is the story of how a species with the most nutritionally demanding and developmentally prolonged infancy of any mammal on the planet managed to keep enough babies alive to be here. Over the course of this documentary, we will move through the full 50,000-year span. We will begin in the first hours after birth, with the specific biological countdown that every woman in the ancient world managed, without any external support. We will examine the practice that researchers now describe as the second arm of infant feeding, one so ancient it appears in the most comprehensive cross-cultural databases. In 63% of households in one documented society, one so biochemically sophisticated that a 2020 for review in allergy and microbiome research found it may have provided irreplaceable immune calibrating benefits to infants, and yet one that has generated exactly 68 peer-reviewed publications. In the entire scientific literature, over 76 years, 68. That number is not an accident. We will examine the evolutionary biology of why the human female body was specifically designed to survive past reproductive age, a feature unique among the great apes, and what that longevity was built to accomplish in terms of infant nutrition. We will enter the coldest decade of the last glacial maximum, when average temperatures across Europe dropped 6 degrees Celsius, and every nursing mother's caloric situation became a genuine emergency that required community-level solutions. We will trace animal milk from its earliest dangerous appearances in unsterilized clay vessels to the genetic mutation spreading through European populations approximately 7,500 years ago that eventually made adult dairy consumption safe. And we will sit with what the bones of prehistoric children have been quietly telling us for decades about when, how, and at what nutritional cost their mothers weaned them. Some of what you hear tonight will feel personally resonant in a way that is difficult to locate precisely. Researchers who work in this domain describe a particular response from women who encounter these findings for the first time, something between recognition and grief. The sense of seeing for the first time something that has always been present but never been named. The intelligence of what prehistoric women built to keep babies alive is staggering. Its invisibility in mainstream historical accounts is staggering in a different way. Both of those things are worth sitting with, whether you are following this closely or letting it wash over you as you drift toward sleep. This story is equally generous to both kinds of attention. The evidence is patient. It has been waiting in permafrost and skeletal collagen and the lipid residues of ancient clay for a long time. If you fall asleep somewhere in the middle tonight and return tomorrow, everything you need will still be here. If you drift off during a chapter and return to it refreshed, you will find the threads waiting for you exactly where you left them. Wherever you are listening from tonight, whatever the hour, whatever the quality of the dark around you, you are part of a quiet, global community of people who come to these ancient stories at the end of the day. We are glad you are here. This is the story of how prehistoric mothers fed their babies before reliable, safe nourishment existed. It is, in the deepest sense, the story of why you are alive. The moment of birth is also the moment the countdown begins. Not abstractly, biochemically. A newborn human arrives in the world, carrying approximately 36 to 48 hours of glucose reserves accumulated before birth. When those reserves are spent, the infant requires external nutrition, the biology that is supposed to provide it. The nursing mother's body does not produce adequate mature milk for 3 to 5 days after delivery. What it produces in those first days is colostrum, a thick, yellowish, intensely concentrated fluid present in volumes that seem almost insulting to a hungry newborn. On the first day, a mother might express 5 milliliters per feeding, less than a teaspoon. By day 3, that might rise to 30 milliliters, just over to tablespoons. The infant cries. The infant roots and searches. The infant, by any behavioral measure available to a caregiver without modern medical instruments, appears to be starving. It is not starving. Not yet, and not quite. Colostrum is not inadequate. It is precisely calibrated for what a newborn immune system needs. In those first days, its composition bears no resemblance to mature breast milk in its purposes. It is loaded with secretory immunoglobulin A, the antibody class that coats the mucous membranes of the newborn gut and respiratory tract, creating a passive immune barrier against the microbial world the infant has just entered for the first time. It contains lactoferrin, which binds iron away from pathogenic bacteria and simultaneously acts as an antimicrobial agent. It contains leukocytes, white blood cells, in concentrations that dwarf what will be present in mature milk. It contains growth factors that stimulate the maturation of the intestinal lining, preparing the gut for the life of digestion ahead. Colostrum is not food in the caloric sense. It is immunological infrastructure delivered in the form of nutrition. But none of that was visible to a woman sitting in the dark 40,000 years ago, watching her newborn cry. What was visible was the crying. What was visible was the smallness of what her body was offering. What was audible was the urgency of the hunger, and what she had available. The honey gathered weeks ago. The animal milk brought to the birth site by the older women present. The sweetened water prepared by her own mother was right there, immediately, offering the appearance of satisfaction. The infant would quiet. The infant would accept the sweetness and stop crying, and the woman's understanding of what had just occurred would be entirely, consequentially incorrect. This is not a story about ignorance. It is a story about the limits of what observation can reach. Without instruments, the colostrum that these women believed was harmful, believed so consistently across ancient Egypt, Rome, medieval Europe, and dozens of documented traditional societies, was described by physicians and midwives across these cultures as too laxative, too thick, indigestible, or simply too strange in color and consistency to be trusted. The Ebers Papyrus of approximately 1550 BC, one of the oldest medical documents in existence, mentions a remedy being spat into a child's mouth by a mother. An early record of prelacteal supplementation as medical practice. Cyrenus of Ephesus, writing in the 2nd century AD, advised mothers explicitly not to nurse in the first 2 days after birth, a recommendation that circulated through European medical literature. For nearly 1500 years afterward, the Islamic tonic ceremony, in which a religious figure or elder rubs premasticated dates or honey on the newborn's palate immediately after birth, predates the oldest written records of it and continues in practice today. Modern immunohistochemical studies have found that this practice may increase expression of interleukin-12 and CD8 T lymphocytes in the neonatal palatal mucosa, meaning the ceremony that cultures understood as a blessing may have been providing measurable immune benefit through mechanisms no one in those cultures had the tools to observe. What this convergence across cultures reveals: the honey, the sweetened water, the premasticated dates, is not widespread ignorance. It is a universal human behavioral response to the gap between birth and adequate milk, a gap that every mother in human history has navigated. The substances varied. The impulse was identical. Fill the baby's mouth with something. Keep the baby quiet. Provide something that feels like nourishment, because the thing that is nourishment has not yet fully arrived. The women who attended births in prehistoric communities were not simply providing emotional support. They were the medical system, and the most critical thing they provided was not any substance at all. It was knowledge. The knowledge of how to stimulate a developing milk supply, positioning, frequency of nursing attempts, the particular handling techniques that could coax colostrum into a reluctant infant, the knowledge of how to recognize when a newborn was genuinely in physiological distress, versus operating within the normal range of hungry but not failing, the knowledge of how to supplement carefully, without undermining the nursing relationship that would need to sustain the infant for the next 3 to 5 years, the knowledge of which prelacteal substances were safer and which were genuinely dangerous, because not all of them were equal, and the difference mattered. Here is what researchers found when they looked more carefully at what determined infant survival in those first 3 days, and it reframed the entire history of what made the difference. The infants who survived were not the ones who received the most nutritious prelacteal food. They were the ones whose mothers had the most experienced women present at the birth. The knowledge held by those women accumulated across decades of attending births nursing their own children, watching milk supplies fail and recover, watching infants thrive and struggle, was the decisive variable, not any particular substance. The knowledge of how to use whatever was available in a way that kept the nursing relationship intact, kept the infant nourished through the gap, and kept the mother's body on track toward the milk supply that would determine the next 3 years of the infant's nutritional life. That knowledge lived in the bodies and minds of the oldest women in every group. It was transmitted by presence, by demonstration, by the quiet authority that comes from having done this before, and having watched others do it wrong. Your own body holds a trace of this. The first foods you received, colostrum if you were, breastfed, formula if you were not, in whatever combination, calibrated your immune system's baseline during the most receptive developmental window it will ever have. The microbial populations that colonized your gut in those first days and weeks were shaped by what you received and who prepared it. The women who were present in your first days of life, whoever they were, whatever they knew, were operating in a chain of transmission that stretches back to the first women, who understood that the first 3 days were not a gap to be filled randomly. They were a threshold to be crossed carefully. Stop and consider what a human mouth actually contains. Not in the abstract, chemically. Salivary amylase, the enzyme that begins the hydrolysis of complex carbohydrates before food reaches the stomach, converting starches into simpler sugars the infant gut can absorb. Lingual lipase, which initiates fat digestion. Immunoglobulin A in its secretory form, the same antibody class that dominates colostrum, present in every milliliter of saliva produced by a healthy adult. Lactoferrin, lysozyme, an enzyme capable of cleaving the cell wall bonds of gram-positive. Bacteria, a natural antimicrobial agent present in every bite of pre-chewed food a mother passes to her child. Growth factors, cytokines, the entire calibrated, immunologically vetted oral microbiome of a woman who has spent decades in the same environmental exposure context as her infant, carrying the same pathogens, producing the same antibodies against them, with a microbiome that has been tested by the same climate, the same water sources, the same seasonal food supply. Everything in her mouth has passed her body's immune surveillance. It has been found acceptable. It has been, in the most literal sense, pre-approved when she chews a piece of meat, a handful of nuts, a tuber pulled from the ground and roasted in the embers of the fire, and transfers that pre-chewed, pre-digested, saliva-saturated food bolus to her infant's mouth. She is not simply softening food for a baby who has no molars. She is performing a biochemical transfer of extraordinary sophistication. She is providing iron in its most bioavailable heme form. She is delivering salivary enzymes that will assist an immature digestive system that produces its own amylase at below adult levels until approximately 2 years of age. She is introducing the infant's developing gut microbiome to the specific bacterial communities of her own oral cavity, communities that, for a child growing up in the same environment as its mother, are the most relevant and most beneficial. Early microbial exposure available. She is building the immune architecture of a child who has no other way to receive this calibration. Researchers call it premastication, the pre-chewing of food before delivery to an infant. A 2020 for review published in a major allergy and immunology journal. One of only 68 peer-reviewed papers on this topic in the entire scientific literature over 76 years. Synthesized what evidence exists on its potential benefits and risks, and concluded that the case for premastication as a beneficial immunological practice had been systematically neglected in favor of its documented risks. Those risks are real. A caregiver with active oral infections, untreated gum disease, or blood-borne illness can transmit pathogens through this practice. But for a healthy woman in a healthy community, the review's authors concluded the benefits: microbiome transfer, immune system calibration, enzymatic predigestion, iron delivery from pre-chewed meat, warranted far more serious scientific investigation than they had received. The cultural footprint of this practice is enormous and poorly mapped. In a comprehensive analysis of 119 cultures, in the most extensive cross-cultural ethnographic database available, researchers identified premastication in 38 of them. Among Han Chinese families in one large study, 63% of households reported the practice. In a 2013 survey of more than 1,900 American families, in the Special Supplemental Nutrition Program for Women, Infants, and Children, between 11 and 17% of caregivers overall reported pre-chewing food for their infants. And among non-Hispanic Black caregivers specifically, that percentage was 42.5%. Among indigenous Semane communities. In the Bolivian Amazon, the practice was documented as standard infant feeding procedure. Among communities in South Africa, in Laos, in the island cultures of the Pacific, in populations across sub-Saharan Africa, this is not a marginal curiosity. This is one of the most widespread infant feeding practices in human history, and the scientific community has produced one paper per year on it. The Avicenna reference is worth pausing on at length, because it is more revealing than it first appears. Ibn Sina, known in the Western world as Avicenna, was writing in approximately 980 to 1037 AD in what is now Uzbekistan. In his 5-part Canon of Medicine, considered the definitive medical text of the medieval Islamic world, and used in European universities through the 17th century, he described premastication as the appropriate method for introducing solid food to an infant whose first teeth had appeared. He specified that premasticated bread should be the initial solid food offered. He was not describing a folk practice to be cautioned against. He was providing clinical guidance in the most authoritative medical text of his era, which means premastication was, in the 11th century, mainstream enough to be codified by the greatest physician of the age, the Ebers Papyrus, recording a mother spitting a remedy into a child's mouth. In approximately 1550 BC, pushes the documented history further back, and the ethnographic distribution across every inhabited continent pushes the actual practice back much further than any document. Researchers who have examined the evolutionary logic of premastication argue it likely predates Homo sapiens as a distinct species, tracing back to the common ancestors of humans and chimpanzees. Some primate populations show proto-premastication, Behaviors. Our lineage may have been practicing some version of this for millions of years before we developed the cognitive capacity to reflect on it. The iron dimension of premastication requires specific attention because it connects directly to the most consequential nutritional crisis of Paleolithic infancy. A newborn human is born with iron stores accumulated during fetal development, enough to sustain normal hemoglobin production for approximately 4 to 6 months. After that threshold, the infant's developing body requires dietary iron to continue building red blood cells, supporting neurological development, and sustaining the rapid organ growth of early childhood. Breast milk contains iron, but in amounts that are specifically designed to be adequate in conjunction with those fetal stores, not independently sufficient past the 6-month mark. In the Paleolithic world, where iron-fortified cereals, iron supplements, and the entire modern system of dietary supplementation did not exist, the primary source of additional heme iron available to a transitioning infant was meat, specifically pre-chewed meat, red muscle tissue from hunted animals, premasticated by the mother into a form a toothless infant could swallow, was almost certainly the most critical nutritional supplement available during the weaning transition, the most frequently premasticated food in every cross-cultural survey, without exception, is meat. That consistency is not coincidental. It reflects a convergent solution to the same nutritional problem, arrived at independently across dozens of cultures over thousands of years, by women who could not have articulated the biochemistry, but clearly understood the outcome. Scientists are genuinely split on how to weigh the risks of premastication against its benefits. In the context of prehistoric populations, those who emphasize the risks point to documented cases of pathogen transmission, bacterial infections, herpesvirus, and in modern contexts, blood-borne viruses. Those who emphasize the benefits point to the observation that premastication has persisted through the full course of cultural evolution across dozens of societies, and that, as one researcher noted directly, if premastication poses significant dangers from the perspective of disease transmission, it is unlikely that it would have sustained through the course of cultural evolution. The evidence is genuinely in tension. What do you think a prehistoric community's practical calculus would have been? An infant developing iron deficiency anemia at 7 months of age, with the cognitive and physical consequences that entailed, against the risk of pathogen transmission from a healthy mother. In a community with no other iron source available, that calculation was made repeatedly across human history. The fact that we are here suggests how it was generally resolved. There is a fact about human women that evolutionary biology has never fully normalized because it has never fully explained, until recently, stated plainly. In modern hunter-gatherer populations that have been studied across multiple continents and multiple methodological frameworks, more than 80% of women survive past menopause. Not a majority, more than 80% among lions, among baboons, among the overwhelming majority of large social mammals. Only a few percent of adult females outlive their reproductive years. Among chimpanzees, our nearest genetic relatives, with whom we share approximately 98.5% of Our DNA, the pattern is similar. A female chimpanzee's body ages and fails in close proximity to the end of her reproductive capacity. The machinery of living, in most species, runs on the fuel of continued reproduction. When reproduction ends, the fuel runs low. In humans, that equation is different, profoundly, structurally different. A human woman's body is built at the level of cellular repair mechanisms, cardiovascular durability, immune function maintenance, bone density preservation, to keep living for 20 to 40 years, past the point at which she can produce children. This is metabolically expensive. Every year of postmenopausal life requires resources that natural selection would not allocate, unless the payoff was commensurate. The question evolutionary biologists have been asking for decades is, what is the payoff? The answer began to emerge from the Tanzanian savanna in the mid-1980s, where anthropologist Kristen Hawkes and her colleagues from the University of Utah were conducting fieldwork with the Hadza people. They arrived expecting to confirm the standard model of the era, that men's hunting was the central engine of prehistoric human nutrition, that the big game protein men brought back was the variable that determined child survival and, by extension, the trajectory of human evolution. What the data showed was something else entirely. They watched the grandmothers. Hadza grandmothers, women past reproductive age, women with no nursing infants of their own, women who, on the standard model, had no obvious survival relevant role in the group, foraged for an average of 4 hours per day during the dry season. That was not a small contribution in terms of raw caloric output. Hadza grandmothers were among the most productive foragers in the entire community, and the food they collected was not going to themselves. It was going to their daughters' children. When a Hadza grandmother increased her foraging effort, the weight and growth of her grandchildren improved measurably. When she was absent or less active, the children in her daughter's household showed the nutritional consequences. The correlation was statistically robust and replicated across multiple field seasons. Hawkes published her initial findings in 1997 in the journal Current Anthropology, proposing what became known as the grandmother hypothesis, that the distinctive post-reproductive longevity of human women evolved specifically because postmenopausal grandmothers made a sufficient contribution to infant and child survival to drive selection for the genes underlying longer female lifespan. A woman who lived 20 or 30 years past her own last reproduction and used those years feeding grandchildren would leave more copies of her longevity genes in the next generation than one who died at the end of her reproductive years. Even though the longer-lived woman was producing no new children herself, her body was functioning as a food security system for the children already born. And the evolutionary math of that contribution was large enough to reshape the entire trajectory of female aging in our species. A 2025 review revisiting the grandmother hypothesis, published in a major evolutionary biology journal, found the hypothesis robust across multiple new lines of evidence and multiple population studies. The core finding holds: human female post-reproductive longevity is evolutionarily connected to infant nutritional survival in a direct, measurable, causally defensible way. The grandmother body is not a biological afterthought. It is a specifically engineered infant survival technology. But the evolutionary mechanism tells only part of the story. What the mechanism required to operate was something more specific. The knowledge the grandmother carried. And here, the Hadza data opens onto a dimension of prehistoric infant feeding that has no analog in any other species. A Hadza grandmother foraging for hours per day in the dry season was not simply collecting calories. She was deploying decades of accumulated ecological knowledge, which underground storage organs were densest at this time of year. In this soil type, at what depth, requiring which extraction technique, which plant's starchy cores could be roasted to a consistency an 18-month-old could manage with the few teeth they had, which fruits, which seeds, which preparations were safe for a child in the weaning transition, and which required processing to remove naturally occurring compounds that would cause harm. The caloric output of a grandmother's foraging was inseparable from the knowledge that directed it. You could not replace her contribution by giving a 20-year-old more hours in the field. The 20-year-old did not yet know what the grandmother knew. This dimension, the knowledge dimension of the grandmother contribution, is the one that the standard archaeological record cannot recover. We can measure the evolutionary consequences of grandmother longevity in living hunter-gatherer populations and model backward from that data. We cannot excavate the specific knowledge that made grandmother longevity evolutionarily valuable, but we can infer its scope. We can infer that a woman who had spent 40 years in a specific environment, watching infants succeed and struggle, attending dozens of births and dozens of difficult weaning transitions, knew things about infant nutrition in that specific ecology that no one younger in the group possessed. That knowledge was the human species' primary quality control system for infant feeding. It was held in women's bodies. It died with those bodies if it was not successfully transmitted. And the evolutionary pressure that kept those bodies alive was, in a meaningful sense, the evolutionary pressure of the knowledge they contained. Consider the grandmothers in your own lineage, the particular competence that seems to arrive with age around infants and children, the specific the knowledge that is difficult to explain, but impossible to dismiss, not sentimental, not instinctive in the simple sense, but accumulated, dense with observation, calibrated by decades of watching what actually works. That competence has an evolutionary history that is specific and measurable. It is not coincidence. It is the residue of 100,000 years of selection for exactly this. In any group of 40 people living in the African savanna or the European steppe 50,000 years ago, something would happen on average every few years. A woman would not survive her labor. The best estimates that by archaeologists and paleodemographers can construct from skeletal evidence, comparative data from pre-industrial populations, and modeling of physiological constraints in the ancestral environment suggest maternal death rates in the range of 1 to 3% of births under prehistoric conditions. Not extraordinary, not the most common outcome, but frequent enough that in any community spanning a generation, every adult in that community had witnessed it. Every woman who had given birth knew it was a possibility she had navigated. Every older woman in the group had almost certainly attended a birth that ended this way and left behind it if the mother died after delivering, was an infant viable, healthy, possibly, hungry, certainly, without a mother? What happened to that infant was the most acute test of the community's infant feeding knowledge system, and the answer, the specific answer that kept enough of those infants alive to matter demographically, was allomaternal nursing. The term means precisely nursing by a female who is not the infant's biological mother. It is documented across human cultures. It is present in the records of ancient Greece, Rome, medieval Europe, early modern China, across the entire ethnographic record of contemporary hunter-gatherer and subsistence communities. Wet nursing, the formal, often commercial arrangement by which a lactating woman nurses another's infant, is its institutionalized descendant. But its deeper roots are biological, not commercial. They are cooperative. Sarah Blaffer-Hrdy spent decades building the theoretical framework that explains why allomaternal nursing is not aberrant human behavior, but its baseline, her cooperative breeding model, developed through the 1990s and early 2000s, and most fully articulated in her 2009 book on human evolution, argues that the defining feature distinguishing human infant development from that of all other great apes is not our brain size, not our tool use, not our language capacity, but our reliance on multiple caregivers for infant survival. Chimpanzee infants are nursed exclusively by their biological mothers. Gorilla infants are nursed exclusively by their biological mothers. Orangutan infants are nursed exclusively by their biological mothers. In what is the most prolonged exclusive nursing relationship in the primate world, human infants, alone among the great apes, evolved in a context where the biological mother was not and could not be the sole provider of nutrition and care. The evidence for this is in our developmental schedule. Human birth intervals are shorter than those of chimpanzees, despite our infants being far more dependent for far longer. The only way that arithmetic works is if multiple caregivers share the load. The research published in Current Anthropology in 2014 examined allomaternal nursing in humans across cultures and found the practice sufficiently widespread to constitute not an emergency behavior, but a structural feature of the human child care system. The Kangsan people of the Kalahari Desert, among the most extensively studied forager populations in the world, practiced what field workers described as kiss feeding, a form of direct oral food transfer from adult to infant that functioned simultaneously as nutritional supplementation and social bonding, and which extended to women who were not the infant's biological mother. As a normalized expression of communal childcare, the intimacy of that act, mouth to mouth, saliva shared, a woman feeding another woman's child with the same directness she would feed her own, reflects a social organization of infant nutrition that our contemporary Western context makes unfamiliar. In the context in which it evolved, it was unremarkable. Allomaternal nursing, actual milk sharing between lactating women, requires a specific physiological condition. The assisting woman must be currently lactating, or must have been lactating recently enough that reinduction of milk production is possible. Lactation can be reinduced in women who have stopped nursing within approximately 6 months. through consistent nipple stimulation. This is not a medical intervention. It is a physiological capacity that evolved in a context where it needed to be available. The question of whether prehistoric communities understood this mechanism explicitly or simply practiced it and observed its outcomes is unanswerable. What the evidence of its widespread presence in living hunter-gatherer societies suggests is that it was practiced, and that it was practiced with sufficient frequency that communities which had this capacity available retained a meaningful survival advantage over communities that did not. The developmental implications of Huddy's cooperative breeding model extend far beyond infant feeding specifically. If the distinctively human social capacity for reading other minds, what cognitive scientists call theory of mind, if our unusual levels of inter-individual empathy and cooperation, if our capacity for language itself all evolved in the context of an infant care system that required multiple caregivers to be attuned to a non-verbal infant simultaneously, then the cognitive architecture of being human is downstream of the same selection pressure that made allomaternal nursing evolutionarily necessary. The woman who nursed another woman's orphaned infant was not performing a selfless act in evolutionary isolation. She was participating in the social structure that made human intelligence possible. Your body carries direct evidence of this. The neurological response that human females, and to a significant but lesser degree, human males experience when exposed to a crying infant, the involuntary attentional capture, the physical discomfort of an unresolved infant cry, the specific pull toward action. This response is not universal among mammals. It is characteristic of cooperative breeding species. It evolved in organisms where the functional response to infant distress by non-parents was a real determinant of infant survival. The fact that you respond, that most women reading this have felt the involuntary response to a crying baby, even when they had no personal relationship with that child, is a behavioral expression of a neural architecture built over hundreds of thousands of years by the survival pressure of communal infant care. You feel it because your ancestors acted on it, and when they acted on it, infants who would otherwise have died lived instead. ** 20-5,000 years ago. If you could have stood on the ground that is now northern France or central Germany and looked at the world around you, you would not have recognized it. The forests were gone, not thinned, gone, replaced by open, wine-scoured steppes stretching to the horizon in every direction. Cold, dry, and in the winter months, almost entirely empty of the plant foods that modern foragers take for granted as a year-round resource base. The Greenland ice cores that allow researchers to reconstruct the climate of this period with remarkable precision show that average annual temperatures across Europe during the last glacial maximum were approximately 6 degrees Celsius below. Present 6 degrees is not a small difference. It is the difference between a temperate mixed forest with year-round plant food availability and a paraglacial environment where most plant foods ceased to be nutritionally accessible for 5 to 7 months per year. It is the difference between a nursing mother with a reliable caloric income and a nursing mother facing a seasonal emergency that recurred every single year of her child's infancy and toddlerhood. The 500-calorie daily surplus required For adequate lactation does not flex. It does not accommodate good intentions or extraordinary effort when the food is not there. When a nursing mother's caloric intake falls significantly below that threshold, her body responds by prioritizing her own survival over milk production. Milk volume decreases. Fat content decreases. The infant, if nursing is the primary food source, receives less than it needs. The consequences accumulate, slowly at first, then not slowly. Weight gain falters, development slows. The immune system, which depends on adequate nutrition to function, becomes less effective at precisely the season, winter, when pathogen exposure is highest. The Paleolithic women who navigated this seasonal emergency every year for the duration of their children's nursing years were not doing so without resources. They had the layered community system we have been examining, the grandmother's underground tubers, roasted and mashed, sustained the transitional-age toddlers who no longer depended entirely on breast milk, freeing the nursing mother's body to concentrate what production it had on the youngest infants. Premasticated animal protein from the winter hunts, lean, frozen, thawed near the fire and chewed to softness, provided the iron and caloric supplementation that breast milk alone could not maintain. The Allomaternal nursing arrangements that distributed the physiological burden of milk production across multiple lactating women in the group reduced the load any individual woman carried. But none of those accommodations could fully address the coldest winters, the years when game was scarce and the hunters returned empty. The years when the underground storage organs the grandmothers relied on had frozen too deep to dig. The years when the entire caloric economy of the group fell below the combined requirements of its members, and someone in the group was going to suffer a nutritional deficit. And the question was who? This is the context in which the Venus figurines become legible as something other than aesthetic objects. Between approximately 35,000 and 22,000 years ago, the full span of the last glacial maximum and its lead-in, the Upper Paleolithic populations of Europe produced a distinctive category of small portable sculptures, almost exclusively depicting women. These were not cave paintings. They were carved, transportable objects, small enough to hold in a closed fist, made from limestone and ivory and bone and fired clay. Many of them depict women with pronounced fat deposits, large, pendulous breasts, a substantial abdominal rounded contour, full thighs and upper arms, wide hips carrying the visible adiposity of someone who is, by any clinical measure, significantly overweight. For most of the 20th century, archaeologists and art historians interpreted these figures as fertility symbols, as goddesses, as expressions of male sexual desire, as ritual objects. A 2020 study published in the journal Obesity proposed a different interpretation that has since generated significant debate in the field. The researchers argued, using extensive comparative data on body fat, caloric reserves, and reproductive function, that the figurines' bodies did not represent an artistic ideal. They represented a physiological survival ideal during recurrent episodes of glacial famine. A woman with the body fat reserves depicted in these figures could sustain pregnancy and milk production significantly longer into a period of caloric emergency. than a woman with a leaner build. The difference in infant survival between a mother who could maintain adequate lactation through the 6th week of a food scarcity event and one who could not was not trivial. Over the span of a population and a generation, it was decisive. The study's authors noted one further detail that is difficult to dismiss. The figurines made during the warmest, most food-secure intervals of the period showed relatively leaner female bodies. As the climate warmed and food became more available, the depicted body type shifted. The figurine that was being carried, displayed, possibly revered, represented the body most needed at the time. In the coldest, hungriest decades, that body was the one that could keep a baby alive through winter. What the glacial context makes visible, with terrible clarity, is that the community's survival system for infant feeding was not a stable achievement. It was a continuous performance under pressure, requiring every component to function simultaneously. When even one component failed, when the grandmothers were depleted, when the premastication knowledge had been disrupted by disease or displacement, when the Allomaternal network was stressed past its capacity by too many nursing infants and too few lactating women, the entire load fell onto individual mothers who were already operating at or beyond their caloric limits. That is what infant mortality in the Paleolithic looked like at its most fundamental level. Not ignorance, not indifference, a system under greater stress than it was built to sustain. In a season colder than any living person in the community had experienced before, the communities that survived the last glacial maximum were, on balance, the communities whose female knowledge and support systems were most resilient. That is the evidence the demography supports. That is what the survival, read carefully, actually says. Asterisk someone made the first decision, and we do not know who she was. At some point, almost certainly before any ceramic vessel existed to hold the evidence, a woman who could not adequately nurse an infant, or whose infant had lost its mother, looked at the animal in front of her, the goat, the auroch, the ewe that had recently given birth, and made a connection. The animal was producing milk for its own young. The milk was there. The infant was hungry. The logic was direct, even if the consequences were not. The earliest material evidence for this decision dates to approximately 5000 BC. Small clay vessels, some with spouts, some with handles scaled for small hands, appear in Neolithic archaeological contexts across Europe, associated in the most evocative cases directly with infant burials. These are not adult vessels repurposed for children. Their proportions, their spout configurations, their scale are all specifically designed for the feeding of a small child. Possible infant feeding vessels made from clay first appear in Europe. In the Neolithic, becoming more common during the Late Bronze Age between 1200 and 600 BC. By the time of the Bronze Age assemblages, the vessel forms had become notably elaborate, animal shapes, with the spout positioned where an infant could approach in a posture resembling nursing, four-legged bodies, small heads, deliberately designed interfaces between the vessel and the infant's face. Someone thought about this. The design reflects an understanding of infant feeding psychology. of the specific resistance an infant shows when asked to accept a new food delivery mechanism, of the behavioral accommodation required to make the transition successful. These are not hastily produced objects. They are the product of knowledge, observation, and considered craft. In September 2019, researchers from the University of Bristol published findings in the journal Nature that confirmed what the context had long suggested. They analyzed the lipid residues preserved inside 3 infant feeding vessels from Bronze Age and Iron Age burial sites in Bavaria and Austria, vessels that had been buried with children, found in the specific archaeological context of children's graves. The residues were consistent with ruminant milk, cow, sheep, and goat, not human milk, animal milk, given to infants in purpose-built delivery devices at sites dating to roughly 3,000 to 3,500 years ago. Earlier, Neolithic vessels from sites across Europe, some dating to approximately 5,000 BC, showed comparable residue patterns when analyzed with similar methods. The practice of feeding animal milk to infants using purposely manufactured vessels was, the evidence shows, thousands of years older than the writing that would eventually record it. Now consider what those women could not have known: the gene variant that allows adult humans to continue producing the enzyme lactase after weaning, lactase persistence, the mutation that makes dairy consumption comfortable for the majority of Northern Europeans, and for populations across East Africa and the Middle East, where pastoralism developed independently, began spreading through Central European populations approximately 7,500 years ago. Before that point, the genomic record is clear. Adult humans, essentially without exception, stopped producing lactase after the weaning transition. The same process that occurs in every other mammal occurred in humans. The milk-digesting enzyme shut down after infancy because, in the evolutionary context prior to dairying, there was no milk to digest past that point. Lactose, the primary sugar in all mammalian milk, reaches the colon undigested in adults without lactase persistence, where it is fermented by bacteria, producing gas, cramping, and diarrhea of varying severity. Large amounts of animal milk consumed by a lactase non-persistent adult produce substantial gastrointestinal consequences. Infants are different, critically different. All human infants produce lactase in abundance. Their entire nutrition in early life depends on it. The lactase gene in infants is fully expressed and highly active. An infant can digest the lactose in animal milk. The infant gut has the biochemical machinery for it. What the infant gut does not have, what no human gut had in any era before pasteurization, is protection against the bacterial populations naturally present in raw, unpasteurized ruminant milk at room temperature. Brucella, Listeria, Salmonella, the full complement of pathogens that modern food safety standards exist specifically to address. A Bronze Age ceramic vessel containing raw goat milk, warmed by proximity to a fire, partially emptied and refilled over the course of a day. That vessel was a pathogen incubator of the first order. The researchers who published the 2019 findings noted this explicitly. The use of unsterilized animal milk in infant feeding vessels almost certainly contributed to the infant mortality of these communities. The vessels they found were in children's graves. The children for whom these vessels were made had died, and yet the practice spread. The vessels multiplied. By the late Bronze Age, they were production objects, made in quantity across a wide geographic area, sufficiently common that their archaeological distribution implies widespread, normalized use. What does it say about the available alternatives that animal milk in an unsterilized clay vessel was preferable? It says the alternative, an infant with no food at all after a mother's death, or during a winter famine that compromised milk, supplied past the point of adequacy, or in the extended gap following a difficult birth, was reliably worse than the risks the vessel represented. That calculation was not made in ignorance. It was made in the specific knowledge that some infants fed animal milk survived, while all infants denied any supplemental nutrition during a crisis died. The surviving infants validated the practice. Their survival taught the next woman what to do. The clay vessels are the physical residue of a knowledge chain, a chain of women watching outcomes, comparing results, transmitting the conclusions. Scientists remain genuinely divided on when sustained dairying exerted enough selection pressure to drive the lactase persistence mutation to its current frequency. The question is whether the primary driver was adults drinking milk comfortably, or infants being kept alive by animal milk supplements long enough to grow up and pass on whatever protective variants they carried. The genomic evidence, the demographic evidence, and the timeline of dairying's geographic spread are all consistent with both hypotheses, and neither is conclusively demonstrated. The honest answer is that we do not know. What we know is that animal milk entered infant nutrition as a survival technology, that it did so under conditions where the risks were real, and the alternatives were worse, and that the knowledge of how to use it, which animals, which preparation, which delivery vessel, which infants were most likely to benefit, and which were most at risk, was developed and transmitted by the same women whose bone spoons and clay vessels we continue to excavate from children's graves. The answer was hiding in the incremental layers of ancient teeth, not in a way anyone could access until the analytical chemistry caught up with the question. But the question had been there, written in enamel and dentin, for thousands of years. How long did prehistoric mothers actually breastfeed their children? Not how long ancient texts recommended, not how long cultural norms suggested. How long did the biochemistry of the feeding relationship last, measured in the permanent physical record of the tissues that were forming during those months and years? Stable isotope analysis of ancient skeletal remains is the method that finally made the question answerable. The principle is precise and elegant. Nitrogen exists into stable isotopic forms. Nitrogen-14, the most common, and nitrogen-15, present, in smaller proportions. The ratio of these to forms shifts predictably through food chains. Each step up the food chain increases the nitrogen-15 to nitrogen-14 ratio in body tissues by a measurable amount. A nursing infant receiving breast milk from a lactating mother is effectively one trophic level above that mother's dietary proteins. The infant's nitrogen-15 signature is elevated, measurably, consistently, in a way that shows up in the collagen of bones, and the mineral matrix of developing teeth. As the infant weans and begins consuming the same foods as other community members, the elevated nitrogen-15 signature begins to decline, falling toward the community's dietary baseline over the months and years of the weaning transition. By sampling the incremental layers of a developing tooth, layers that form month by month during childhood, preserved permanently in the enamel and dentin, researchers can reconstruct the feeding history of an individual child with month-by-month Resolution study of late Holocene hunter-gatherers from Lake Salatra in Patagonia, Argentina, found an early incorporation of supplementary solid food between 9 months and 2 years of age, combined with a late cessation of breastfeeding at approximately 5 to 6 years. 5 to 6 years, not months, years. The introduction of complementary foods began in the first year. The same premasticated proteins and processed plant foods we have been examining, but breastfeeding continued alongside those supplements for years afterward. A 2-year-old in this population was receiving both breast milk and supplementary foods. A 4-year-old may still have been nursing occasionally. A 6-year-old may have nursed on demand in times of illness or stress long after being primarily self-fed. That pattern, documented through isotopes in skeletal remains and consistent with what ethnographic observation of living hunter-gatherer populations has recorded, represents the natural weaning schedule of our species in its ecological baseline condition, a gradual, years-long transition rather than a discrete event. Compare that to what the isotopic record shows for early agricultural populations. A Bronze Age study examining weaning practices across 5 archaeological sites in Europe. Populations that had been farming for several generations, keeping domesticated animals, producing grain-based foods, found a very different pattern. Supplementary feeding began at approximately 6 months. Complete weaning occurred around 2.6 years, roughly 2 years earlier than the Patagonian hunter-gatherer data. That compression of the weaning window was not biologically inevitable. It was ecologically driven. Agricultural communities had food available in more regular, predictable quantities than forager communities, which made early weaning practically possible. But they also had social and economic incentives to shorten birth intervals. More children meant more labor on the farm, and the hormonal contraceptive effect of extended breastfeeding was a direct constraint on fertility. Earlier weaning meant shorter birth intervals meant more pregnancies per woman per decade. The agricultural economy had a demographic demand for more children that the biology of extended breastfeeding was limiting. The nutritional cost of this transition was written in the enamel of the children who lived through it. Enamel hyperplasia are visible defects in tooth enamel that formed during periods of significant nutritional stress or illness during childhood. Appears at elevated rates in early Neolithic and Bronze Age skeletal assemblages, compared to the preceding Mesolithic hunter-gatherer populations. The children of early European farmers show more enamel hyperplasia than the children of the foragers who preceded them in the same regions. That pattern is consistent with the isotope data showing shorter breastfeeding duration. Children weaned earlier, transitioning to a diet based primarily on cereal grains and occasionally supplemented with animal products were not receiving the same density or diversity of nutrition that their hunter-gatherer predecessors had received from a longer breastfeeding relationship combined with diverse supplementary foods. The Cispical region of Siberia, studied using stable isotope analysis across multiple archaeological periods, shows a particularly instructive pattern. During the early Neolithic period, when populations in this region were forager-fisher hunters with access to diverse, high-protein food sources, infants were breastfed until approximately to 2-3 years, supplemented with fish and animal proteins from approximately 6 months. During a later period characterized by greater subsistence uncertainty and population stress, breastfeeding duration shortened, and supplementary feeding became more grain-dependent. The bones of children from the stressed period show elevated markers of nutritional inadequacy compared to the more affluent forager period children. The feeding practices that produced the best outcomes, longer breastfeeding, protein-rich supplementation, diverse food introduction, were not the practices that were easiest to maintain under stress. They were the practices the ancestral system had been specifically built to sustain. The picture emerging from this converging body of isotope data is not one of prehistoric mothers who were doing something simple and natural that modern life has complicated. It is one of prehistoric mothers operating a sophisticated, community-supported nutritional system that was producing outcomes by the measures of child skeletal health, growth markers, and nutritional adequacy that early agriculture actually degraded before eventually, over millennia, recovering to a different kind of adequacy. The hunter-gatherer infant feeding system was, for the individual child, nutritionally superior to what early agriculture offered. The trade-off was scale, agriculture's nutritional costs per individual child were offset by its ability to support population densities that no forager system could match. The individual child paid the cost of the transition that benefited the civilization. Asterisk, there is a category of knowledge that leaves almost no trace in the archaeological record. Stone projectile points survive for hundreds of thousands of years in the right conditions. Bone tools survive for tens of thousands. Ceramic vessels survive for thousands. Even the organic residues inside ceramic vessels, the lipid molecules that confirm what the vessel held, survive long enough for modern chemistry to read them. But the knowledge of which plants, prepared in which way, at which stage of the plant's annual cycle, using which processing technique, could sustain a child between 6 months and 3 years of age through a difficult winter. That knowledge lived in women's minds. It lived in the movements of their hands. It was transmitted by standing next to someone who knew and watching what they did. Not once, but dozens of times, until the knowledge became procedural memory rather than declarative memory. It left no residue, almost none, ethnobotanical research. With contemporary hunter-gatherer and subsistence communities has recovered fragments of what must have been a comprehensive and highly localized knowledge system. The Hadza of Tanzania, whose subsistence ecology researchers believe offers a reasonable analog for the environmental pressures that shaped much of human prehistory in sub-Saharan Africa, offer the most extensively documented case, frank, Marlowe's long-term study of Hadza subsistence found that the average adult Hadza woman gathered approximately 3,000 calories worth of food per day. The backbone of this was underground storage organs, tubers, corms, bulbs, roots. Specifically, starchy underground structures that the plant uses to store energy through the dry season. Concentrated enough in calories to make excavation energetically worthwhile. Soft enough after roasting in coals or ash to be consumed by an infant with minimal dental equipment. The Hadza's primary plant food during the dry season is the tuber of Vigna frutescens, known locally as equa, which requires digging to depths of 30 to 50 centimeters in hard laterite soil, with a wooden digging stick, a labor-intensive extraction that experienced Hadza women perform with remarkable efficiency and that younger women take years to master. The same elder women who most efficiently located and extracted these tubers were the ones observed most consistently provisioning grandchildren the knowledge of where to dig, at what time of year, in what soil type, at what depth, knowledge. That determined whether a foraging trip returned 3,000 calories or 300 was specifically held by the oldest women in the community and transmitted informally to daughters and granddaughters over years of shared foraging. Time Dotha, Kangsan people of the Kalahari Desert have been studied in comparable depth. The Mungongo nut, Shinziophyton rotanini, forms the caloric backbone of the Kung diet in the dry season, dense in fat, protein, and calories, storable without processing, mechanically demanding to open in a way that requires specific tools and techniques, and specifically associated in the ethnographic record with older women's food preparation and distribution. Kung women roasted, cracked, and prepared mungongo nuts as a staple supplementary food for transitional-aged children. The specific preparation for infant-appropriate consistency, cracking the shell, extracting the kernel, dry roasting to a specific degree, and then grinding or pressing into a paste, required knowledge that was neither obvious nor quickly acquired. It was held, primarily, by older women who had been preparing these foods for decades. Across the Ache people of Paraguay, the Agda foragers of the Philippines, indigenous communities throughout the Amazon basin, and forager populations studied. Across sub-Saharan Africa, researchers have documented analogous patterns. Older women's specific knowledge of plant-based infant foods, representing a critical nutritional technology that was not intuitive, not easily improvised, and not readily transferable except through sustained direct transmission. The convergence across these geographically distant and ecologically distinct populations is too consistent to be coincidental. It reflects a convergent evolutionary and cultural solution to the same problem. The gap between what breast milk alone could provide and what a growing infant needed during the weaning transition, filled by plant-based foods whose identification, preparation, and appropriate use were held as a specific domain of female elder knowledge. Here is where the history of paleoanthropology requires honest examination. The discipline spent most of the 20th century analyzing and celebrating the stone tool record, the projectile points, the hand axes, the scrapers and burins associated with hunting and butchery. This was not methodologically neutral. Stone tools survive, They are visible in the record, and they were, for most of that century, assumed to be the primary domain of male activity. The organic tools associated with women's foraging, digging, sticks, bark containers, woven carrying bags, leaf surfaces for food preparation, decompose without trace. The grinding stones associated with plant processing, when found at all, were long categorized as peripheral or secondary to the hunting. Implements that dominated the interpretive framework, the entire domain of plant-based infant food preparation, the most nutritionally critical female knowledge domain in the entire prehistoric record, left almost nothing that the standard archaeological toolkit was equipped to find or recognized as significant. Researchers in the emerging field of the archaeology of childcare, scholars who specifically investigate the material record of infant and child survival rather than adult tool use and subsistence, have begun to redress this. But the recovery is partial. What the ethnographic evidence from living forager communities allows us to infer about the depth and sophistication of prehistoric women's plant knowledge is far richer than what the material record alone would suggest. The underground storage organs that Hadza grandmothers excavate, roast, and pass to toddlers today are the descendant foods in the same ecological category, prepared by the same learned techniques, distributed through the same grandmother to grandchild, provisioning pathway that sustained the weaning infants of Paleolithic Africa for tens of thousands of years before agriculture existed to replace them. Wherever you are tonight, if you find yourself knowing things about feeding children that you did not formally learn, if the impulse to offer particular foods in particular forms to a struggling child arrives with a certainty that feels older than your own experience, you may be touching something very old indeed, not mystical, transmitted through the women who were present in your own early years, and through the women who were present in theirs, in a chain of practical knowledge that stretches back through time further than any institution that has ever claimed to manage it. In December 2019, researchers published a study in PLOS One that identified something no one had looked for before. In a collection of objects that had been sitting in museum storage for years, the objects were bone spoons, specifically spoons manufactured from cattle bone, recovered from the early Neolithic site of Grad Star-Savo in Serbia, and associated with a settlement context dating to approximately 6,500 years ago. They were not large spoons. They were small, with shallow bowls and short handles, and their surfaces bore a set of marks that scanning electron microscopy revealed as having been made by human teeth, specifically by the small, blunt, incompletely erupted teeth of a child, primary teeth, baby teeth, in the process of coming in. The bite pattern was characteristic of a child between approximately 8 and 18 months of age in the teething phase, who had the spoon placed repeatedly in their mouth while someone used it to deliver food. These were baby spoons, not culturally symbolic objects, not tools used primarily by adults. Baby spoons, made from cattle bone, found in a domestic food preparation context, bearing the tooth marks of the infants they had been used to feed, recovered from a settlement 6,500 years ago. The study's authors identified them as among the earliest direct physical evidence of spoon-feeding infants in the archaeological record, and their manufacture from cattle bone, a material that is a byproduct of animal domestication, places them precisely at the intersection of the two transitions that most dramatically reshaped infant feeding in human prehistory, the beginning of settled agricultural life and the beginning of sustained animal domestication. The bone spoon sits at the visible edge of something much older and entirely unrecoverable. The practice of spooning food to an infant, of using any implement to transfer pre-prepared food from a preparation surface to an infant's mouth, is older than cattle bone and older than any material that would survive. A flat stone, a large, leafed finger, which requires no tool at all. The oldest baby's spoon in the world is the first finger that transferred pre-chewed food to a waiting infant mouth, sometime in a prehistory so deep that no date can be attached to it. The Serbian bone spoon is simply the oldest version that happened to be made from something durable enough to last. The proliferation of infant-feeding vessels across prehistoric Europe traces a trajectory that mirrors the pressures of agricultural life. Small spouted clay vessels, some with handles, some with animal shapes that invited an infant to approach the spout, in a posture resembling nursing, appear in Neolithic contexts. From approximately 5000 BC, a 2021 study examining a large assemblage of these vessels from sites in Central Europe found that their frequency in the archaeological record increased significantly during periods of greater population density and urbanization, as communities grew, as birth spacing shortened, as the economic demands of agricultural production competed with the time-intensive requirements of prolonged breastfeeding. The material technology of infant supplemental feeding expanded proportionally. The vessel was not replacing knowledge. It was the material expression of knowledge. The knowledge of what to put in it, when to use it, which infant needed it, and how to use it without causing the bacterial contamination that raw animal milk in an unsterilized ceramic container at ambient temperature could produce. The animal-shaped vessels deserve specific attention. Across the Central European assemblage, Germany, Austria, the Czech Republic, Croatia, a recurring vessel type appears, a small ceramic container modeled in the form of a four-legged animal, with a spout positioned at what would be the animal's mouth or at an equivalent position that allows an infant to approach the vessel. In a suckling posture, the detail that these vessels were often made in the shapes of cows, goats, and sheep, precisely the domesticated ruminants whose milk was being placed inside them, was not coincidental. Someone designed this with specific knowledge of infant behavior, the resistance of a transitioning infant to an unfamiliar feeding mechanism. The way an infant accustomed to nursing will more readily accept a vessel that mimics aspects of the nursing posture. The design is a piece of applied developmental psychology executed in fired clay. Approximately 3,000 years ago, the lipid residue analysis that the University of Bristol team published in 2019 confirmed the contents of 3 such vessels from Bavarian and Austrian burial sites. The residues were consistent with ruminant milk, cow, goat, sheep. The vessels were found in children's graves, found there, presumably, because the people who buried those children understood that the vessel had been central to the care those children had received, and that burying it with them was the appropriate thing to do. The grief embedded in that choice is specific and material. A woman had filled that small animal-shaped vessel, had held it to an infant's mouth, had tried. The infant had died anyway. She buried the tool of her attempt with the child she had tried to save. The care and the loss are inseparable in the archaeological record of these objects. What the full trajectory of infant feeding technology, from bone spoon to clay vessel to animal-shaped drinking cup, makes visible is the gradual materialization of knowledge that had previously existed. Only in practice, every new tool that appears in a record represents accumulated observational learning that has reached sufficient density and reliability to justify manufacturing a durable object to implement it. The bone spoon represents the knowledge that soft food delivered by implement to an infant's mouth was nutritionally valuable and practically achievable. The clay vessel with a spout represents the knowledge that liquid foods, animal milk, porridge, ground seed preparations, could be delivered in a controlled quantity to an infant who could not yet manage a cup. The animal-shaped vessel represents the knowledge of infant feeding psychology, specific enough to inform the design of an object intended to reduce the behavioral resistance of a transitioning baby. Each of these objects is a document, not a text. Document, a knowledge document. The intelligence encoded in these objects belongs to the women who made them, used them, refined their design through observation, and taught the next woman in their community how to use them. None of those women's names are recorded. The objects remain asterisk knowledge of this kind, practical, embodied, life or death in its consequences, does not survive casually. It survives through relationships, specifically through the relationships between older women and younger women who are preparing to become mothers. It survives through co-residence, through shared domestic space, through the years of proximity in which a daughter watches a mother manage an infant's feeding, transition and builds, without necessarily being taught explicitly, a procedural understanding that can only be acquired through witnessing it done correctly many times. Before you need to do it yourself, it survives through the births attended before your own. The specific knowledge of those first 3 days, of how to read a struggling nursing relationship, of when a supplement is needed and what kind and how much, that can only come from having been present at other births, not just your own. It survives through the particular kind of teaching that happens between a grandmother, who is still active in a community, and the daughters and granddaughters who are watching her, observing the food she collects and the preparations she makes, and the specific decisions she makes about which child receives what in which season. All of that requires continuity. The chain does not merely weaken when it breaks, it severs, and what was held in it cannot be recovered from the next generations. Improvisation. A young woman whose mother died before she was ready to have children of her own. In a community where the knowledge was held by individual women rather than spread redundantly across multiple elders, faced the same biological problems that the knowledge existed to address, and faced them without the resources the knowledge provided. The infant mortality of those situations is not in the individual birth record. It is in the demographic pattern of communities that lost their elder, women prematurely, and in the skeletal health records of children who grew up in communities where the nutritional knowledge chain had been interrupted. This is a dimension of prehistoric infant mortality that the standard archaeological framing of individual birth and death misses entirely by archaeologists working in the emerging field of the archaeology of childcare. Researchers like Sheen Halcrow of the University of Otago, whose work specifically examines this skeletal and material evidence for infant care practices, have pushed the discipline toward exactly this kind of systemic analysis. The question is not only which individuals died in infancy. The question is what determined the survival differential between communities. What made some groups better at keeping babies alive than others in comparable environmental conditions? And whether that differential reflects differences in individual maternal biology or differences in the collective knowledge and support infrastructure that surrounded each birth. A 2021 study that reframed the archaeological interpretation of prehistoric infant mortality rates made a point that deserves emphasis. The assumption that had dominated the field for decades, that approximately 40% of all prehistoric babies died within the first year of life, was based primarily on the frequency of infant skeletons in burial assemblages. But burial assemblages systematically overrepresent infant deaths. Living children who survived did not leave equivalent physical markers. The visible record is the mortality record. The survival record is invisible. The researchers argued that this asymmetry had led to a systematic underestimation of how effectively prehistoric mothers cared for their infants, that the care was, on average, better than the death record implied, and that the deaths that did occur were more likely to reflect specific circumstances of failure, rather than the baseline performance of the system. What this means for the story we have been telling is significant. The layered system of female infant feeding knowledge, premastication, grandmother provisioning, allomaternal nursing, plant food preparation, was not a system operating at marginal efficacy, barely keeping enough infants alive to sustain the population. It was a system performing well enough that its failures were the exception rather than the rule. That the mortality it could not prevent was the residue of its limits, rather than the measure of its capacity. The successes are invisible in the archaeological record. The children who received pre-masticated meat at 7 months and grew into healthy adults, who had their own children and grandchildren, left nothing comparable to the bone spoon buried in an infant's grave. They are the unmarked majority. They are the reason the species continued. The transmission chain for this knowledge had a specific vulnerability that became more acute as human populations transitioned from mobile forager groups to settled agricultural communities. In a mobile forager group of 30 to 60 people, the knowledge was distributed across multiple women of different ages who lived in constant proximity. A young mother had access to her own mother, to her mother-in-law, to the older women of the group, to the women who had attended her birth and would attend her child's weaning transition, all at close range, all available to consult and observe. The knowledge was redundantly distributed. The loss of one elder woman was serious, but not catastrophic. because others held overlapping knowledge. In a settled agricultural community of several hundred people, that structure of intimate female knowledge networks became harder to maintain. Extended family proximity was disrupted by marriage patterns that moved women away from their own mothers' communities. The larger population created enough social distance that the intimate observational transmission became less reliable. The knowledge began to be formalized into written medical texts, into midwifery manuals, into the professional expertise of wet nurses and healers, who held it institutionally rather than familiarly. Formalization preserved some of it, but formalization also filtered it, retained what the literate, predominantly male medical establishment recognized as important, and discarded what it did not recognize or did not value. The plant-based infant food traditions, the premastication practice, the full ecology of grandmother provisioning, none of these made it into the canonical medical literature of the ancient world in any form that recognized their systemic importance. They were women's practices. They were passed woman to woman, quietly, in the spaces between the things that history was recording. What you have heard tonight, across these chapters, in the research that grounds each of them, is not a reconstruction of a lost paradise or a simple narrative of female victimhood. It is the recovery of a knowledge record that was always there, that was always consequential, and that the institutions of historical scholarship systematically failed to investigate. Because the people who held it were women, and the domain in which they held it was the domestic and reproductive sphere, that those institutions consistently regarded as beneath documentation. Recent science is recovering it, slowly, through stable isotope chemistry and lipid residue analysis and ethnographic fieldwork and the archaeology of childcare, recovering what the record allows, inferring what the convergent evidence from living cultures permits, building, piece by piece, the picture of what the women who kept this species alive actually did and actually knew. That work is not finished, but it is underway, and what it reveals, with every new study and every new analysis, is consistently more sophisticated and more consequential than the silence around it had ever suggested. We began tonight with a gap, 3 to 5 days between a newborn's arrival and the arrival of adequate milk. A small biological interval that, seen from the outside, looks like a design flaw in a species that would have struggled to survive it. But the gap was never the problem. The gap was the entry point. It was the biological fact around which an entire knowledge system had been built, tested, refined, and transmitted across 50,000 years of human survival. A system so sophisticated and so robust that its absence or disruption was among the most reliable predictors of infant mortality. In the archaeological record, the answer to the question this documentary asked, how did prehistoric mothers feed babies before milk was safe, is not simple. It is layered, like the system itself. They fed them with pre-masticated meat and nuts and roasted tubers, delivering iron and enzymatic predigestion and microbiome transfer through a practice that has appeared in 63% of documented households in one culture and 42% in another, and been studied in 68 papers. In 76 years, They fed them through the specific, evolutionarily grounded contribution of postmenopausal grandmothers who foraged for hours a day to provide the calories that nursing mothers could not simultaneously produce for both themselves and their milk. They fed them through allomaternal nursing, the redistribution of the physical burden of lactation across multiple women in a community, whose neural architecture had been specifically calibrated over hundreds of thousands of years to respond to infant hunger regardless of biological relation. They fed them with plant foods prepared by women who held ecological knowledge so localized and so specific that it could only be transmitted by standing next to someone who knew, watching them do it for years before it became your own. They fed them, eventually, with animal milk in unsterilized clay vessels, accepting the bacterial risk because the absence of any nutrition was reliably worse. And they did all of this in a climate that, during its coldest periods, reduced the caloric arithmetic of lactation to a genuine emergency, requiring every component of the system to function simultaneously. That system was not fragile in the way. Individual women were fragile. Individual women died. Individual milk supplies failed. Individual communities faced years when the grandmothers were ill. The game was scarce. The underground tubers had frozen to deep. The system survived those failures because it was redundant, because the knowledge was distributed across multiple women of multiple ages, because the evolutionary pressures that shaped it had selected for exactly this kind of collective resilience. Over tens of thousands of years of experience with exactly these kinds of failures. The system bent under maximum stress. It did not break. If it had broken entirely, if the redundancy had been insufficient, if the knowledge chain had been severed comprehensively, we would not be here to discuss it. What you carry from this story is written into your biology in ways that are specific and, in several cases, directly measurable. The architecture of your immune system was calibrated in the first weeks of your life by the specific microbial communities it encountered, communities whose composition was shaped partly by what was transferred to you through the feeding methods used in your infancy, and partly by the women who were present in those first weeks, and the specific environments those women inhabited. Your gut microbiome, now understood to be foundational not just for digestive health, but for immune function, metabolic regulation, and neurological development, was partly established by those earliest feeding experiences. The neurological wiring that generates your response to infant distress, your capacity for extended empathy with vulnerable others, your inclination toward gathering and communal problem solving in situations of shared vulnerability. These are behavioral expressions of neural architecture that evolved in the context of the cooperative infant care system that kept early humans alive. And there is the fact of postmenopausal longevity itself. If you are a woman past the age of 50, the fact that your body is built to keep living, that the evolutionary machinery of your cells is oriented toward decades of continued function, past reproductive age, is the most direct biological evidence that exists for what older women's contributions to infant survival have meant to this species. You were not built to fade. You were built to keep going, and the evolutionary reason for that The specific selection pressure that shaped it over hundreds of thousands of years was the nutritional contribution that older women made to the most vulnerable members of the next generation at precisely the moment those members needed it most. Modern life has built a different infrastructure around the same biological problems. Formula, pasteurization, sterilized bottles, lactation consultants, neonatology units with equipment that can sustain infants born before the 30th week of gestation. These are extraordinary achievements. They have saved millions of lives, but they are also, in the deepest sense, successors to what prehistoric women built. The same fundamental project of ensuring infant survival during the most vulnerable period in the most nutritionally demanding species on the planet, pursued with the best available technology of each era. The women who filled clay vessels shaped like goats with raw milk 3,000 years ago were doing the same thing that the neonatologist adjusting a feeding tube does today. The best available answer to this same ancient question, the question, has not changed, only the tools. What changed, and what the full scope of this documentary has traced, is who held the knowledge. For 50,000 years, it was held by women, transmitted between women, validated by the outcomes of women's observations, and sustained by the evolutionary architecture of the female body itself. It was held outside institutions, outside writing, outside any of the systems that received the name of knowledge in a formal historical record. It was held in exactly the place that formal history has consistently failed to look: the domestic, reproductive, communal space where women were managing the survival of the generation that would come after them. That knowledge is not gone. It is transformed, distributed differently, held in different forms. But the chain from those earliest women to the women who are alive tonight has never been broken. Every woman listening to this is the surviving proof of a system that worked. The biological and behavioral residue of 50,000 years of accumulated intelligence about how to keep the youngest and most vulnerable human lives intact through whatever the world offered. That intelligence is in you, not as memory, as architecture, as the specific shape of everything you are. The women who built it did not know their names would never be recorded. They did not need to. They were not building for legacy. They were building for the child in front of them, in the dark, in the cold, with whatever they had and whatever they knew. Rest quietly tonight. Everything they carried forward arrived. It arrived in you. The chain holds.